Okay, so maybe I can just start by reiterating what everybody's been saying for the last few days. I want to thank the Sophion team and also Amgen for putting this great conference together, and also particularly to Duncan, who is running around organizing things for us, for doing such a great job and bringing everybody together, and such great speakers, actually. It's been really impressive. So my lab's interested in a number of different types of ion channels. We're interested in GABA receptors. We get more interested in looking at voltage-gated sodium channels. But what I thought I'd do is tell you about something that we've had a long-held interest, which is to look at ionotropic glutamate receptors. And I was going to tell you about some recent work that we've done on that, which kind of came as a surprise to us. And I'm going to try and give you a bit of a spoiler alert before I get into the data, so you can start to think about what I'm saying. So for anyone who's unfamiliar with ionotropic glutamate receptors, they basically represent the major excitatory neurotransmitter receptor in the central nervous system, which means they're involved in almost everything. So from vision, auditory system, learning and memory, you name it, ionotropic glutamate receptors involved in that. And so just to try to capture some sense of where they're located, they're found in these synapses, on the postsynaptic density. And the two kind of brother glutamate receptors that sort of team up there are the AMPA receptors, which is what I'm going to be telling you about today, and also NMD receptors. And you can see it's packed full of different types of proteins, which over the last 20, 30 years, people like Hugener and a whole bunch of other people have dissected the roles of these different kinds of proteins. The reason I put this scheme up is for the reason, if you think about all the talks that have sort of been presented, even the last talk, is that what you tend to think about this, and I had this same bias, is that when you have an AMPA receptor in a synapse, which opens and closes within a few milliseconds when being activated, you tend to think about it in terms of the activation process, is that nothing really happens until the neurotransmitter receptor comes along, and then all the action happens, right? And so what I'm going to tell you is that that's probably not the case for AMPA receptors. What's happening at AMPA receptors is that they're predetermined to respond to the way in which they respond to neurotransmitter, and also to pharmacology. And I think that's something that pharmacologists really need to start to think about when they're designing drugs towards glutamate receptors. So before getting to that, I'm just going to give you a quick kind of overview of ionotropic glutamate receptors and AMPA receptors. So involved in many, many different types of physiological processes. So perhaps not too surprisingly, they're involved, here's the 4 different subtypes of ionotropic glutamate receptors. They're involved in a whole slew of different types of CNS disorders. And I've just come up with just some of the CNS disorders that have been identified. If anybody's been paying attention to Nature in the last month or so, there's been 3 papers showing that AMPA receptors actually seem to be heavily implicated in this very aggressive cancer called glioblastoma multiforme, where the cancer cell forms synapses, literally forms synapses with neurons, and sort of mimics itself as if it's a synaptic transmission. And that seems to be very important in driving the proliferation of the cancer. So given this, not surprisingly, there's a lot of need to try and understand just the structure function of these receptors. And there's been a lot of success by a number of groups, primarily spearheaded by Eric Goh, who came up with this structure. The first author of this work was Sasha Savlevsky. He's got his own group now in Columbia. And what they found is, and I'm going to kind of zip through their data very quickly, is that this is an AMPA receptor, so it's a homomeric tetramer in this particular example, but they're usually heteromeric in nature. So these are made up of 3 distinct domains. There's a transmembrane domain, which makes up the core of the receptor. And then right in the middle is the kind of what we think of being the business end. There's 4 binding sites that form these kind of clamshell or Pac-Man-like structures that sort of crunch down on glutamate, and that is thought to trigger the whole activation process. And then there's the amino-terminal domain that seems to be important among a number of things, but also in the assembly of these receptors. So these different types of ionotropic glutamate receptor family all seem to form this similar type of architecture. There's some little twists and turns, but basically they all have that same kind of architecture. Another thing that's important to remember about them is, in addition to these 3 distinct domains, this is actually a pretty big protein. it's 100 angstroms, at least 100 angstroms. So that means that it reaches almost all the way across the synaptic cleft. And so there's some emerging ideas that maybe that causes interactions with proteins on the presynaptic side. And the data I'm going to tell you about will give you a clue to why we think that may be important as well, right? And they form this dimer of dimer interactions. So there's actually two ligand binding domains here that form back to back. And it turns out that's a really important thing. So one thing that hasn't really been resolved until the work that we started to get into is an observation that had been made looking at both AMPA and kainate receptors, which are another type of glutamate receptor, and they're closely related to each other. And this is sort of schematic of a little cartoon, is that when the cryo-EM structures, they found with the cryo-EM structures of AMPA and kainate receptors, and they looked at the amino-terminal, then they found that there was quite a bit of disparity in the structures of the amino-terminal. Nobody really understood what that really meant. It was static structures. All they knew is that they could define them into different types of sort of architectural anatomy, if you like. And nobody really had a good explanation to why that was. So it turns out we ended up getting ourselves involved in looking at this, but we didn't think we were looking at that at the time when we started the experiments. It started off in a very simple way, and it was a grad student in my lab who kind of followed up from some work that I had done some time ago, back in 2002. And what I noticed, so it was very simple, kind of doing some biophysics of these AMPA receptors. And what we found is that when you look at the desensitization properties of these receptors, so just to orientate people in terms of the experiments, we're pulling outside-out patches and doing extremely rapid exchange of the agonist solution, so it mimics synaptic transmission. You get a rapid activation of these receptors, and if you change it with different types of halide ions, you can see you can change the amplitude of the response, and also you can change the time course of desensitization of these receptors. In contrast, if you only done a 1 millisecond pulse, so basically all you're really doing is primarily activating the receptor and pulling off the agonist before the channel gets a chance to desensitize, then the anions don't have any effect on that. So we had this anion effect that seems to be very selective at regulating desensitization. We thought that was something that would be interesting to pursue. The other kind of interesting thing that came out of it is if you looked at the ionic radius of the anion, then the degree of the rate of desensitization accelerated the bigger the anion you got. And that made us think that it was probably some distinct binding site that this anion was binding into. It also affects the recovery of desensitization. I'm not going to tell you about that given the limitations of time. So given that, because we thought, well, these anions are probably binding to some site, we teamed up with the Jette Kastrup's group at the University of Copenhagen, and we asked her to take the isolated, just to sort of orientate you, she basically made an isolated crystal structure of the ligand binding domain here, and then basically try to crystallize and use bromide ions, because bromide ions have a particular diffraction pattern in the crystal structures, so she could identify where the anion pocket was. And lo and behold, she actually found these bromide ions, which are shown here in more detail. They're actually in this dimer interface, so this interface between these two ligand binding pockets right in there, right? And the thing that piqued my attention from work that I had done more than 20-odd years ago with Mark Mayer when I was a postdoc in his lab, is that there was a serine 775, which seemed to be kind of close to this binding site. And most of you are probably not familiar with this, but the serine 775 is actually part of an alternatively spliced part of the AMPA receptor. All the AMPA receptor genes undergo alternate splicing through this process called the flip-flop cassette, which is summarized here. So the flip-flop cassette, which is found between the TM3 and the TM4, it's part of the ligand binding pocket. I'm going to show you in a second where that is structurally. But the serine, it's either a serine if it's a flip, or it's an asparagine, which is found in a flop. And what I had shown 25 years ago in a Neuron paper that Mark and I had found is that that single amino acid residue had a profound effect on the ability of AMPA receptors to respond to positive allosteric modulators. And at the time, we didn't really have much more of an explanation, not simply a description, right? So anyway, so that piqued our attention. And just to give you a little bit of understanding from a structural perspective, so here's now, this is the flip-flop cassette here. So what I've got shown here of these amino acids here, they actually form this kind of structure, this alpha helical structure that's got a kink in it. What was really interesting is that the 775 residue was right at the sort of apex of this kink. So it might be ideally positioned to be doing some sort of functional things that we hadn't really understood. Over the years, a number of other people, so this is the paper that I had with Kathy Parn and Mark Mayer way back, first identifying this, but then a number of people had shown that just the AMPA receptor by itself, if it was a flip or a flop, then they have different gating properties. And then again, the 775 was implicated into this. And then Ingo Gregor's group primarily had shown that the flip-flop cassette, so this same part of the protein, which makes up the ligand binding domain, seemed to be very important in the trafficking of AMPA receptors and their exit out of the endoplasmic reticulum. So the way we've seen it at the time, and I think the way the whole field's seen it at the time, is you get these three disparate type of things, and for some reason, they're all targeted through this, this one kind of structure. So there's one on this side, and if you look carefully on the light blue, there's one on the other side. So there hadn't really been some sort of unifying explanation to why you have this flip-flop cassette. So to try to understand that, now going back to this allosteric regulation by anions, we said, well, why don't we just look at flip-flop and see their sensitivity to different anions. And as we anticipated, what we found with the flip, so this is GluA2 flip receptors, right, which have a serine there, you can see that they're very profoundly affected by different types of halide ions which are there. Whereas if you look at the flop, so this is the wild-type flop, so just a difference in this one amino acid, then you can see that, first of all, the kinetics are much, much faster, and anions almost don't have any effect. And that was really curious to us because we were consulting with Jette back in Copenhagen when she had the structures of both the flip and flop, then the anions bound to both of those sites. So there was something that didn't kind of make sense, right? It wasn't like in this case with the flop that the anions weren't binding. They were binding, so there was something that just didn't make sense, right? And then the thing that really made us kind of compelled by the whole thing is if you simply take the serine, which is found in the flip, and replace it with asparagine, then although you don't change the kinetics because you need some other residues to do that, now the anion sensitivity, again, was completely dulled. It was completely attenuated. It didn't go completely, but it was greatly attenuated. So that one amino acid was having a big effect. So what's going on? So back in 25 years ago, when me and Mark Mayer were thinking about this, the way we thought about it is glutamate comes along, and it's only then when the agonist binds that the flip-flop cassette now starts to kick in the allosteric pathway that leads to channel gating. But another possibility, which seemed a bit more intriguing and something that would be interesting to pursue, was basically some of the work that was coming out from Sasha Sobolewski's lab, Eric Guo's lab, and also from Hiro Furukawa's lab, is that when they did these kind of morphs of how AMPA receptors work. So here's the agonist binding. Here you can see there's a closure of these clamshells, but then you can see there's this crunching down of the overall protein. So there's a kind of compression of the protein. And we wondered maybe what happens when the anions are actually binding in here during the process of desensitization. It's actually occurring long before the agonist turns up, so it's actually predetermining how the agonist occurs. So to try to look at that, we actually turned to an old friend of mine, Mike Edwardson, who's the chair of pharmacology at Cambridge University, and he's a real expert in using atomic force microscopy. And basically, with atomic force microscopy, you can make two important measurements. You can look at the height of the protein, and you can also look at the movement of it. And I'm going to show you those two things in a minute. So by just using this probe, this cantilever, you can actually look over fairly brief periods of time with different pharmacological manipulations. You can look to see if there's compression of the protein. So that's what we did. So what you can see on this side of the slide is when you put in, from a control condition, you wash in glutamate, just as you would expect, and also predicted by Sasha and from Eric Guo's work, is you get this compression of the protein. It's about 7 angstroms. So it's about almost a tenth of the height of the protein. So it's pretty substantial amount of compression of the protein. And you get it for both the flip and the flop, although you got different effects depending on the anions which were present. So that was a hint that something might be going on. But the thing that really blew us away was that if you just simply took these single AMPA receptors in these layers, then you can actually get as much, even more so in some cases, as much as 10% of change compression in the height of the AMPA receptor without glutamate even being there, right? So, and then to convince us even further of that, it only happened with flip, but it didn't happen with FLOP. Remember, the FLOP was the AMPA receptor that wasn't very sensitive to anions. Likewise, if you took the flip, but then mutated it, so it would have that asparagine that you would have in the FLOP, then you no longer had this effect. So this one amino acid, which was part of this very important alternatively spliced protein, was actually dictating the response of this. And what was important is this was all predetermined, right? It was happening before the agonist came along. So what might be going on? So what we thought might be going on is because the anions bind to both the flip and flop, right? So it can't be because one version of the protein can bind anions and the other one can't. It must be something else. So we thought it might be due to just the intrinsic kind of nanoscale movement of the protein at rest, so the apostate, in other words. And so we looked at that again using AFM. So we're sort of probing the top of these dimer of dimers of the amino terminal. And I'm going to try and see if these things work. So you can see in the apostate, you're actually getting a fair bit of movement. And then when you put glutamate on, I hope you can see a lot more movement going on. You're able to kind of calculate that. You can look at the control over time, and you see the movement in presence of glutamate. Not too surprisingly, you get a lot of agitation of the protein which is going on. I won't have time to take you through all the movies, but just to give you a sense of what we found when we did that now to the flip and flop. So here in the flip, it kind of behaved itself, so you had this sort of intrinsic moving going on. We put glutamate on, there was much, much more flexibility in the protein. If you put a competitive antagonist, then you could completely kill all of that, suggesting that this was driven by the glutamate. But here's the thing that really kind of surprised us. When you did the flop, then the mobility of the protein was even much greater than what you get with the flip in the presence of the agonist. And then interestingly, the variance in that actually reduced when you put glutamate on, and then you could kill it to some extent by polluting the antagonist. So what it meant is the apostates of the protein were actually fundamentally very different from each other. And that's something that's really surprised me, even though, of course, it's a possibility. It's just that we don't think about it in terms of synaptic transmission. And so just to re-emphasize that, so here's the flip again. Here's the flop. And then the thing that really made it causal to us, if you make that single point mutation and then look at the mobility, you can see that now you've got this high mobility just by changing that one single amino acid in the flip to an asparagine, you now flip, if you can excuse the pun, into behaving like a flop. So we thought that what's going on is that all of these ligands can actually bind. It's just that they're binding to proteins which are moving around much more than the others. And so therefore it's harder or easy for the binding to occur. And that's why you get the functional consequence. So if I was to go back then and look at the stuff that Mark and I had done with Kathy Parton and then other people had looked at, it'd be known that flip and flop have very different decay kinetics from each other, which is shown here. So we argued this is actually predetermined by the nature of the apostate. And likewise with the positive allosteric modulators, with cyclothiazide has a different effect on flip and flop. And again, we think that's due to differences in this apostate. And so just two slides just to quickly finish. So we said if that's really true, then we should be able to then go to auxiliary proteins. So this is proteins which bind to AMPA receptor and change their properties. So there's a number of different types of auxiliary proteins. And if you do it with an AMPA receptor here, arg-stargazin, which is a TARP, or a cornicin, you can see you can profoundly affect these proteins. And what we had shown in a previous neuron paper a couple of years ago is we identified the binding site where the TARPs actually attached to the AMPA receptor. And there's only just a few amino acids. So we argued that, sort of reason to ourselves, is that if these AMPA receptors are moving about at different rates, then the ability of the TARP to interact with the AMPA receptor will be perturbed. And so in the flip, you'll get this great regulation that you see here, but you won't get it in the flop. And that's exactly what we got. So here's the flip, showing here. If you put the flop, it's like there's no regulation whatsoever, even though we've tethered the TARP to that AMPA receptor. So it's the intrinsic movement of the protein before the glutamate comes along. It's determining that. When we do a single point mutation, again, to do that, you can get the same effect. So just in summary, what it really says to us is that unexpectedly, even to me, is in glutamatergic transmission, the makeup of the AMPA receptor is really predetermining how that synaptic response is going to behave. How it's going to behave in terms of its responsiveness to glutamate, its pharmacology as well. And I think that's something very interesting for the pharmacological industry to really think about when they're sort of chasing the activated state of the protein, when maybe they should be thinking about the nature of the apose state. And then, of course, this alternate splicing overrides this very profound effect of these auxiliary proteins. And again, we tie it back in to the nature of this apose state. So if I can just quickly thank the people involved in this. There's a whole bunch of people in my lab, a really talented grad student, Brent Daw, who's now finished, gone on to bigger, greater things. In Mike Edwardson's lab, it was Fahim Katir. And then in Copenhagen, and also with a group in Chile who was involved in the computational aspects of the EFM. So thanks again for your attention.
Thanks, Derek. Great talk. Questions from the floor? So let's go here, and then we'll go to Bruce, and then I'll save mine for after that. Please.
Fascinating talk. Have you done experiments where when you replace, instead of doing the halide exchange, let's say you put in some sort of completely non-conducting, something that is a big anion. I mean, do these go away if you do that? Have you done experiments like that?
Have we done that?
Simple question.
No, I don't think we haven't done that. But so I've only presented half of the paper that we published just recently. And in there, we actually look at heteromeric receptors and the effect of TARPs. And it turns out the TARP stoichiometry adds another level of modulation on top of it. So I just thought, given the group here and the timing of things, I would just tell you about that. But we haven't tried that experiment. But what you would expect that if you have a big anion.
Yeah, I mean, I'm kind of thinking that because it is such a, rather than having an halide, having chloride or fluoride, anything like that in there, I mean, you know, maybe you get a different effect when you look at these sweets.
Yeah, I mean, there's a physical limitation of what can get in there. So my guess is at some point you just won't get any effect. But we haven't tried that, so maybe we should have a crack at it.
The size of the movements is amazing.
Yeah.
Do you think it's possible that in the apose state it ever moves enough to actually get into the open state so that you can, you know, have opening in the absence of...
Yeah, I mean, if you look at Tony Auerbach's work, right, I mean, he has made a very compelling argument in nicotinic channels. It usually involves mutations, and he's developed a very elegant way to explain that for nicotinic channels. And so I chatted to Tony about this. In terms of glutamate receptors, nobody really thinks that that can happen. But given this, you could imagine that might happen, right? It's just whether or not you can detect it, right? So, but it's a surprise to us. You know, we kind of, when you do Markov models, you start off with the resting state, you just assume it's kind of not doing anything. You need to start somewhere. But it turns out that that's the thing that seems to be very important, which is a bit of a surprise.
We have a question over in the back corner. Thanks.
Very interesting talk. So given that the synaptic cleft is probably about 20 nanometers and these movements are pretty substantial and that there are transsynaptic signaling columns that hold things together, can you speculate on whether that a group of AMPA receptors doing this is going to affect those interactions and indirectly affect synaptic activity?
Yeah, so that's a great question. It's something that we're interested in. Unfortunately, a whole bunch of other people are interested as well. Roger Nicole's already started to look at it by replacing the amino terminal of AMPA receptors in the hippocampus. Ingo Gregor's done it. I think Roger had a PNAS paper, an EMBO paper for Ingo. I think there's going to be more coming out of this. You know, that you can understand how that's going to be very important because AMPA receptors cycle in and out during these memory consolidation or at least the cellular basis of that. So you can see how this kind of mechanism may be important, right? So, but yeah, it's early days. Let's see how it plays out.
Okay, great. Thanks so much, Derek.
Cheers.
Good
Thanks.
talk. So the final speaker of this session is Professor Len Kaczmarek from Yale.